And so I thought I'd tell you a little bit about, there's going to be some science in here because I am a professional physician scientist. Uh, let's see, I'll tell you a bit about me and what I do. I'll tell you a bit about, for a professional physician scientist, geographic path and professional path actually kind of become the same thing after a point. But without seeming too wise, because I don't think I'm that age yet, I think I'd like to tell you about some of the stuff I learned, mostly from the mistakes that I made, mostly things that I wish I'd done better, mostly things I wish I'd known when I was in your shoes, and that if you can avoid some of my mistakes, that would be really great. And then I'll also, somewhere in this talk is a picture of Carolyn back in the day. Okay, with me in this plate, actually. All right. So if you were to meet me these days in the wild, you'd probably meet me at the American Society for Hematology meeting, or the Cancer Research meeting, or the Keystone meeting on protein degradation, because I am a professional hematologist. I take care of patients with cytogenetically complicated leukemias, and I also have a little sideline business in allogeneic stem cell transplant. And so that's what I do 4 weeks a year. And the remaining time is mostly taken up with running a lab. And my lab studies something that you might have come across because it has gotten a lot of cachet in the last 10 years, but it's probably just started to make its way out of the niche, which is the field of targeted protein degradation, which is something we'll talk about later, but is this burgeoning field of trying to understand the design of small molecules that can tag onto a protein that you want to go away and direct it to either the lysosome or the proteasome for degradation to create either phenotype in a research program or a therapy against a disease that you might be interested in. That's my headshot. So if you did meet me in the wild, that's my official picture. That's what I try to show people what I look like, which that's the best I can do. So that's all I was able to come up with. The middle picture is actually where my lab is, and this is the lab itself, which is a pretty small lab in an open space building. But that's where I spend most of my time. That's, of course, what you see when you see me. I think, hopefully, the more interesting thing is, how did I get there? And so I'll tell you about that in a second. But this is also, I thought I'd show you my pitch, when I meet people for the first time and I talk about who I am and what I'm interested in. My pitch is, there are, of course, roughly 30,000 genes in the genome. There are presumably that many or maybe a few more proteins. They have lifetimes, so they're made and degraded. Degradation is regulated. And so at some point when you've exceeded your lifetime, when you've exceeded your lifetime, you experience a post-translational modification like a phosphate group or a ubiquitin group or a sumo group or something, some signal that tells the cell to get rid of the protein by directing it to a ubiquitin E3 ligase that ubiquitin tags on the protein, at which point it's degraded in the wood chipper of the proteasome. Not a one-way street, you can reverse all of those steps. This is a dynamic process that's always acting on all of the proteins in your body, but it can be dysregulated in disease and it can be therapeutically exploited. And these are a list of the projects that if you were interviewing for a position in my lab, I would say these are the 7 projects that we do. And we are trying to understand the biology and find drugs for the process. It's pretty, it's a great chapter in my life to be doing science at this level. But this talk was actually a chance to look back and figure out how the heck I got there. Because sometimes, since I'm 45, you look back and wonder. And so here are my folks. Those are my parents in the middle. My father was a Cook County Hospital hematologist studying sickle cell disease, and he had some experience in virology. So he was a clinician with some scientific interests. My mother's a urogynecologist. She became an OB-GYN when she was in her 20s and a urogynecologist in her 60s. So she's a little bit of a maniac for learning things. And they are, really, honest to God, they are my heroes. These are my maternal grandparents. Engineer and lawyer unusual for the 1930s in India, first female lawyer in South India, and a woman of extreme temper, extreme achievement, very handy with throwing objects if you did badly in school, but a real terror. And I really idolize these people. And these are my paternal grandparents. My grandfather in particular was a vet who was for some reason deeply interested in parasitic infections in cattle. And so he has apparently two species of bovine nematodes named after, that he has helped name. He named them after his assistant, but which I think is pretty cool for, again, India in the 50s. But, I come from a family, therefore, of nerdy people. And my mother and father moved to the States and to Chicago here, directly straight from India in 1971. Father was there, resident in hematology at Cook County Hospital. My mother is over here doing obstetrics. I was born shortly after. These are the cutest pictures I could find of myself, and they're, and then that's me in preschool. I would like to say precocious child, but I think I was actually only noticeable for being very smiley, but I think that's about it. But the curveball, and I think where I'd like to start talking a little bit about things that brought me here today, is that my parents took a sabbatical to move back to India from their academic careers. And we moved to India when I was 12. I didn't come back till I was 18. These are the schools that's me up there, if you can tell. Small boy with big glasses, also taller boy with big glasses. And moving to India was a huge, and probably remains one of the seminal cataclysmic events in my life, because it was a big change to go from suburban Illinois to the middle of India, number one. And then number two, it was my first introduction to a schooling system of that nature, which was to say a schooling system that really emphasized memorizing facts, taking tests, being the first student in the class. Being the number one student in the class was the whole ball game. Being the number one student in your city was the next ball game. And taking a national exam after that was the real ball game. And so, for example, during my schooling there, I definitely absorbed a lot of material, I would say. I absorbed a lot of raw material. So if you were to ask me, what is the subspecies of lungfish that lives in the Bay of Bengal? I don't know anymore, but I could have told you then. No problem. Or if you asked me, please draw the reproductive organs of the frog, I would have been, yes, I can do that too. So that emphasis on memorization, emphasis on recall, very, very powerful thing. But a really kind of a grotesque, not to knock it, but it's also, I survived it, but it's a grotesque educational system in that when I was this age up here, for fun, I took that final exam. 150,000 people took that exam that year. You had to come in the first 75 slots to get the prestigious medical school slot. And I came in 2,774, which, out of 150,000 people is not a bad grade, but that rank got me a seat in the secretarial college to become an agricultural manager at a cattle farm in a very back of the woods town in the middle of nowhere. And I always think about that because, I'm on the faculty at Harvard Medical School now. But in an alternate universe, I would be managing a cow farm. And it is a real testament to accident and fate that I am the child of my parents, and my parents were returning to the States, and I was headed to the University of Chicago. I can't take credit for that. I always think that is a really strange, unearned turn of fate. And there's something to be humble about too, because my mother took this exam in 1968, and she came in number one. So there's also a sense in my mind sometimes of lesser son of, greater parents. Okay, so I went to University of Chicago. You guys are from Chicago, so you have no need to be introduced to all this snow. I can tell you, I got off the plane, it was 52 degrees, and I thought I was gonna die of hypothermia. They gave me $400 to move from India. I spent it the first day on an Eddie Bauer coat, rated for the McMurdo Sound expedition to Antarctica. And I couldn't afford to buy another coat, so I wore that coat for all years in college. And people still remember me this day as, oh yeah, the Indian guy who wore the polar coat. And it's just, I got used to it. These quotes are true. These are from my era back then. I have a t-shirt for each of these, which I couldn't find in time for this talk. But, it was really pretty hilarious. But it was also absolutely the perfect time for me because I was a kid who had spent 5 years memorizing every last scientific fact, biology, physics, math, chemistry. I had a huge amount of stuff in my head. When I got to college at the University of Chicago, most of the curriculum was liberal arts. And I stood up in the middle of General Assembly during orientation and I shook my fist and I railed and said, nobody needs to know anything about literature. Nobody needs to know anything about history. We need to study science, mathematics, and invent the future. Why are we doing all of this? And I think, thankfully, nobody laughed too hard. But it changed. The experience was actually it transformed my life, and almost everything I am today, I really owe to that experience. Those are my two best friends. He works for Intel now, and she's a geriatrician at Mayo. But they were calm and sane, and sort of reined me in. This is my academic hero, Carl J. Weintraub. He was a 18-year-old, half Jewish, half Catholic kid hid by a bunch of Quakers in Rotterdam for 3 years during World War II. He is 6'8" tall. He hid in a closet for 3 years during the war that I saw a picture of, and it's about the size of that chair. He just hid in there for 16 hours a day. And he read books, and he taught himself 6 languages during that time. And in 1944, he escaped to New York, and then he was sent to Chicago to become a college student. He stayed for umpteen number of years and ended as the chairman of history. And he ran a invitation-only history class, 16 students. Instead of meeting 3 times a week, it met 6 times a week. It was the most horribly demanding class. He was an extremely terrifying person. He would usually say something like, Mr. Koduri, now that you've fouled the waters irretrievably, maybe someone else could try again. And I thought he hated me. But he taught us a lot about the arc of history. He taught us a lot about why it's important to know something about our civilization, and not for any other reason than it matters for knowing something about our own motivations. It matters for asking yourself in the morning the parallel question, which this is Peter Miller, who is my other life-changing professor, who started his class with the question, in a world where in the Holocaust camps, there were poetry clubs devoted to Rilke, Hölderlin, and other humanist poets. There were music clubs for Mozart, but it didn't matter. There were still gas chambers, and people went to one and then went to the other. What is the use of studying anything? Why do you care about studying history, art, science, or whatever if it doesn't prevent that from happening? And I think those two questions were really seminal to forcing me into a conversation with myself about why on earth I was doing anything that I was doing. And if you want to talk about that later, we can talk about that. But I think I came to a sense of I feel compelled to study not just science, but science and medicine, because I don't want to have a career strictly divorced from these humanities, but I am a scientist at heart, by temperament, by training, but in my soul. But that's what shifted me a little bit towards going into something that my parents actually hoped I would not become a doctor. And so I had to it was like the reverse Indian phenotype. I had to sort of say, no, actually I am doing it. All right, but to being a scientist, the other person I met who changed my life forever is this guy. His name is Malcolm Casadaban. For any of you who are serious, and I mean like serious bacterial geneticists, Malcolm Casadaban is one of the giants he's one of the giants of bacterial genetics of the 70s. And a lot of the logic and the systems that he designed for use in bacteria have since found their way into eukaryotes and mammalian engineering systems. But he was there at the very, very beginning. His PhD was with John Beckwith at MIT. And his PhD is this paper, which, in typical jargon doesn't sound like a lot, analysis of gene control signals in DNA fusions and cloning. But how many people have used like a restriction enzyme in this class? All right. How many people have used a recombinase to flip a gene in? Okay. Malcolm cloned genes before any of that was available. Cloning in the 70s was done entirely with super complicated crosses using bacteriophage. And if you wanted to use a restriction enzyme, you purified it yourself. You grew up 100 liters of bacteria, And there were no epitope tags, there's no column purification. This was, you lysed the bacteria, you purified them on anion and cation exchange columns, you found the exact conditions to purify that one protein at purity out from all the other bacterial proteins, and then you saved them. I was going to say some other word. You saved a lot of it in the freezer. And so, but this paper is really kind of remarkable because we take this for granted. If I showed you a gene and I said, hey, there's some regulatory sequences, there's a promoter, there's some structural genes, this turns on this, makes this, turns into proteins, you'd say, yes, I learned that in high school. But in the 1970s, none of this was known. And you could, if I gave you a piece of DNA and asked you, with the tools that you had then, where's the promoter? Where's the structural part of the gene? Where's the part that says to stop transcribing the gene? How would you know? And this is what he did. This is his his system is, his thinking. And this is, the reason I put it up there is not just for bacteriological trivia of the this is like the worst Jeopardy category ever. This is to tell you something about how he thinks, because a lot of this rubbed off on me. And his idea was, I don't want to solve this question for one gene. I want to develop a tool or a system so that anyone who comes with this question is going to have a tool to plug it in and figure it out. And so the system that they worked out, was revolutionary at the time, was they took this protein LAC-Z, which you might have heard of, right? It's an enzyme that hydrolyzes, in this case, it makes this enzyme beta-galactosidase, which metabolizes lactose. And on the right indicator bacterial plates, white colonies will become blue, or on McConkey agar, clear will become pink, but you can tell when the LAC-Z gene went on. And then again, it's a heroic thing given it's trivial today, but it's heroic 50 years ago, that he made this vector system where you could put the LAC-Z gene in and then you could screen a bunch of these fragments of DNA for whether or not they were promoters. Because if they were really promoters, they would turn on the LAC-Z gene and that colony would turn white or red, depending on what plate you were on. So it was a system for screening it was one of the first promoter traps. And it wasn't, of course, because Malcolm was a profoundly logical man, he said, well, we can do it for the start, why don't we do it for the finish? I'll also do it for termination codon stuff. So I love this because it's an example of, if you're presented with a question, you have an opportunity, you can either be the person who solves the question quickly and moves on, or you can say, I want to build a system to answer this question because there is a generalizable class of that question, which someone's going to want to know. And I like that. I think it's the bigger picture. And I think that's a theme that I really want to return to, is about seeing the bigger picture. Okay. But these are some of the things he used to tell me. These are actual quotes from Malcolm. He died tragically, like a few, sort of 10 years later, for different reasons. But he used to say this a lot: science is not abstract, because I was very prone to saying, I don't understand. He would say, read the paper, do the experiment. It's done by real people like me. He would look at me and say, you could do this. I don't know why we're talking. You could just go do it. And I liked this one. He loved tinkering with instruments. He liked just doing stuff. He always thought doing it badly was better than anything else, except for doing it better, I guess. And then I liked this one, but this is not a quote by Malcolm. This is actually, what I took away from him was also cloning bacterial genes is what Malcolm, or making systems or understanding new recombinase is what Malcolm did with his life. But really, what he showed me is that the modality isn't important. For Malcolm, I thought being a scientist was how he was. It was his approach to also driving his car, or changing the faucets or whatever. It's a way of life. It's a way of being and living. And I think working with him for that time was to be absolutely implicitly, but very clearly ask the question, are you a scientist? Does this resonate with you? Is this how you want to live your life? And I think the last thing I'll say is, for anyone who's thinking of going into science, this stuff matters. Malcolm was tenured, he was super famous, and he was also the crappiest grant writer I've ever seen in my life. As in, he didn't even try. Forget he wrote the grant badly. Amazingly, he somehow missed the part about writing grants, and so eventually the lab closed. It's very hard to close the lab of a tenured professor, but U of C forced him out about 15 years after I met him, and he went to industry and did other things. But it's a reminder to me that it doesn't matter if you're a genius who helped invent modern molecular biology. You still have to write the grants. You still have to pay the bills. You still have to do the thing. And I think that that's a lesson worth heeding. Okay, so I was a biochem major along with being a history major, and the temple of biochemistry is Cambridge in England, the home of so many of the great legendary structural biologists. And I got a fellowship to Trinity, and I got to go to the Department of Biochemistry, and I got to work with my hero, my scientific hero, Max Perutz, who, Austrian-born, British naturalized citizen, aided the British war effort, and spent the better part of 3 decades inventing the technology necessary to obtain high-resolution X-ray crystals. This is the first one published, along with Kendrew, of myoglobin, which is the oxygen-carrying pigment in your muscles. And amazingly, when this structure came out, this was the first protein structure, right? Like up till then, Bragg had shown all kinds of crystals, regular small salt crystals. This was the first protein crystal. He was incredibly disappointed when he saw it. He remarked to his colleagues, it just looks like a blob. It looks terrible. It's ugly. I mean, it's that Greek idea, the truth should be beautiful. Turns out you had to learn to see the beauty in the myoglobin, because what was super cool is we know that myoglobin carries oxygen, but it's not clear how it picks it up in one place and lets it go in another place. And it turns out that there's a really complicated, basically porphyrin ring buried in the myoglobin, and the oxygen, there's actually a histidine that's sort of linking up with the oxygen, and when it's oxygen bound, it's coming in, and when it's oxygen apo, it's letting it go. So I always think that, it's not, we breathe, but really it's the hemoglobin that breathes. You have this oscillation. The histidine is like a little ticker, dropping and picking up oxygen. This is literally the fundamental mechanism of life on an oxygen-rich planet. And it comes from an atomic level understanding of this protein. This is what I wanted to do with my life. When I read this story, this is all I wanted to be, because it was the coolest thing I'd ever seen. And so I went to Cambridge, and then it turns out Max Perutz is retired because he was already like 80,000 years old at this point. And so he was not taking students. But he put me in touch with this guy who became a lifelong mentor. His name is Benjamin Bonaventura Luisi. He's actually American from Pasadena. Bonaventura. But he is actually a really famous crystallographer on RNA binding proteins. And he's also one of the coolest, again, a scientist kind of person. This is just to show you what I did. Most amino acids are handed. We mostly work with L-amino acids. That, in fact, is familiar to the crowd, right? It turns out that L-amino acids mostly form right-handed helices. So, this kind of helix over here. But it turns out that the energetic solutions in the Raman plot for the relevant phi-psi angles and the energy state are such that even if you had an L-amino acid, there's no reason you can't form a left-handed helix. The energetics are the same. And yet there are literally zero across all life forms on this planet, there are no left-handed alpha helices. There are other left-handed helices, but they are, for the structural biology nerds out there, we can talk about those. But no left-handed ones. And the question is, why not? Because you either have to say, somewhere in the dawn of time, some bacteria decided to go right instead of left. That's what we're doing today. Or there's a hidden energetic constraint that we don't know about, that we need to figure out, that is actively constraining protein folds. It's right in front of us, and we don't know what it is. That's how Ben's mind worked. And I think it was such a pleasure to work for him, because these are the kind of things I learned with him. He would always say, being a scientist isn't just about doing the experiments. It's a way of life. It's a way of thinking and seeing the world. He insisted, as in you would be disciplined if you did not put down your pipette at 3 o'clock and go to tea. And I thought, this is the dumbest thing I've ever heard of. I have work to do. Why are we going to tea? I mean, I like tea, but still. And you would go to tea, and you would pick up this little cucumber sandwich or whatever. There's a tomato sandwich, a cucumber sandwich, and an egg sandwich. You would get one of these sandwiches, and you would get your tea, and you would go to sit at the table. And the rule in the department was you couldn't sit with anybody in your lab. You couldn't sit with anybody you collaborated with. You couldn't sit with anyone you'd sat with the week before. You had to sit with someone new. And you would sit with them and say, ah, yes, I've seen you around. I work on blah, blah. What do you work on? And you would learn. And this was Ben's way of telling us, I think, that the whole point was not to like do the experiment, but the point was to step back for a second, at least for a little bit, and learn about your field. Learn about it in the best way, not from a paper, but from other people who were doing it like you. He also had an astronomy club, which I loved. It was optional, so I went, of course, I love astronomy. And, you know, it was a hard physics class. So the first time I heard of Hubble's constant and the red shifting of galaxies and the dark matter paradox was not in an astrophysics class in college. It was in a Master of Biochemistry class at Cambridge, taught by a guy who was not an astrophysicist, but just knew a lot of physics and thought it was really cool. Super helpful. There's a lot of the math that's in that stuff that's come back to help me later on in my own life. I don't think you should do it for that reason. I think the coolest thing about it was, again, being a scientist is a way of life. You don't just do crystallography and then go home and stop being a scientist. It's that you go home and you start wondering, hey, is the universe expanding like a bubble? Is it in a yo-yo thing? Or is it coming back to this? I don't know, but it turns out someone does. And you could spend a little time learning about that to sustain yourself, to feed that hunger to learn things that I think is not so much a hunger as a habit and an ability. And you need to train it and feed it and give it bits to keep it going. Also, I would say, not for nothing, but as a middle-aged man who is not as fit as Carolyn is, and not a runner, exercise. Ben worked out seriously once a week, and I thought it was again, I was like, why would you do that? Now I'm like, got it. I really wish I'd done it. So, all right, so here's the picture of Carolyn, I promised you. I went to Harvard in 1997. I was an MD PhD student, so I did 2 years of medical training. This was my anatomy final. Harvard Medical School was a really ridiculous place at that time. The rule for the anatomy final was you had to come in costume. You couldn't come in your regular clothes. And there was a theme every year, and this theme was apparently drag butler, which I was like, I don't even know what that is. So I came, that's me there with my broom. I brought my broom to the final. It was really amazing. I left University of Chicago. This is sort of my second point. As I left University of Chicago, I had a 4.0 average. I felt very, very smart. I'd gone to Cambridge. I was like, I'm gonna just have a great time. People are just, I'm gonna explode. It's gonna be amazing. And when I got to HMS, I realized that I was probably slightly above average, slightly. And there were, in fact, all the prizes I'd applied for and not gotten, all the guys and girls in this picture had gotten. All the scholarships I'd applied for, all the stuff I hadn't gotten, they had gotten all of them. But no one talked about it. They were there to learn. They were there to do cool stuff. And it's true across the board. It was true with everybody I met. And it was a dawning sense for me that, and I think really important for me, because I was really on the verge of a type of arrogance that I have tried hard to beat out of myself since, and thankfully going to school did do it, was to realize that the world is full of absolutely amazing people, absolutely brilliant people, most of whom know more than you do. And that's not a knock, that's an opportunity to say, how do you go forward? Will Rogers has that quote, never miss a good opportunity to shut up. And that's become like one of my mottos is, listen, people have things to teach you. Remarkable people have things to teach you. And so on that note, this was my PhD advisor, Steve Blacklow, that's what he looked like then. He's actually now lost a lot of his hair, but he's the chairman of biochemistry at HMS, and he's an advisor to some venture capital fund, and he's quite rich. So he's traded youthful skinniness and hair for other things. I want to point out, these are the 3 postdocs I worked with, who remain my friends to this day, who I collaborate with or am in contact with to this day. And I graduated in 2006, so imagine that this is 17 years ago, and I'm fortunate. And so I took my crystallography interest to my new medical world and said, other diseases we can understand with structural biology, and at that time, familial hypercholesterolemia, which it turns out is one of the most common genetic diseases in the world, mostly because there are all kinds of mutations in the low-density lipoprotein receptor that, transports cholesterol from the gut to the liver and sort of around the body. The ligand binding domain, the part of the receptor that binds the lipoprotein, is made up of 7 copies of this protein, which is called an LR. It's an LDLA repeat. The 5th one is most often mutated, and nobody knew why. Number one, and nobody knew why did the mutations cause the disease? So you guys are not in high school, so the disease is a loss of function phenotype. So the question is, how does the mutation cause the LOF? And I think there are certain obvious answers, but at the time, it was really the first example, in some sense, that this disease was a protein folding disease. And so the first paper I wrote was on characterizing these residues that I've labeled are all actually mutated in the disease. And it turns out that this little doohickey of a fold, it's only 40 amino acids, it has no recognizable alpha helix or beta sheet. It's held together strictly with this little cobweb of hydrogen bonds and a 2-residue hydrophobic core, which is, for the protein folding people out there, this is a very different thing from most proteins. But it was cool because you could actually use some cool thermodynamics and cool NMR and cool biochemistry to actually figure out how the fold was specified using the disulfides and some zinc coordinating residues and the hydrophobic core, and you could figure out how the mutations killed the protein. So it helped define familial hypercholesterolemia as a protein folding disease, which is pretty cool. And that was my first paper. I was so amazed. I was like, however, the paper went to biochemistry, which is, I don't know if it's a journal you guys know, but in the world I live in now, right, which is impact factor, mean girl high school, biochemistry impact factor of 8, science is at 45, cancer discoveries at 50 and God knows what. So I felt super proud of myself for having completed the work. And then I sort of said, am I a failure? My paper's only in biochemistry, So I decided I would be very ambitious. So I got to the next project and I said, this one is going to go to Science, Nature, or Cell, guaranteed. And I picked a totally new protein, this LRP6 protein, which is LDL receptor related protein number 6. And it's got all the same pieces as the LDL receptor, but more of it. So it's this gigantic 1600 residue, 4 beta propeller protein that's transmembrane. It's like the white whale of crystallography projects. And I was a first-year grad student who had never crystallized anything larger than 100 amino acids before. But I said, I'm brilliant, I'm amazing, I can do this. Okay. This is actually a lie. 4 years later, with literally no publishable data, 0. I had tried expressing this protein in 10 kinds of bacteria, 3 different strains of yeast, baculovirus, and then 4 different mammalian cell systems. In the end, I was never able to make sufficient quantities of functional protein to work with. And at this point, I was a 5th-year grad student who had 1 paper in biochemistry, and I gotta say it, it was I came across the hard truth. Now, I think I have slides for this purpose, but I'll just tell you, I came across the hard truth that I'm actually pretty average as scientists go, which is an important realization because it's only after you realize that then you start to think, well, what makes an excellent scientist and how do I become that? But I realized I had spent a lot of time bashing my head against, 4 years is a long time. A year and a half should be enough time to say, hey, this is really hard. What am I missing? What something's wrong here, right? But I was convinced that I could do it. And then this paper came out, which it turns out that there is a required protein folding chaperone that you must express in order to make this protein. And in the absence of that gene, you cannot make this protein no matter how hard you try. So all those 4 years of work that I had done were a waste because I didn't have I was not also expressing it with this protein, MESD. Moment of truth time. Do you graduate with the paper you have and say, I just wasted my time? Or do you throw up your hands and say, I'm a failure and leave completely? And I'm really lucky that someone whom I really treasure as an advisor and as a mensch sat me down and walked me through this part, which is instead of doing a structural project, which I came to Boston to do, why don't you do a functional project? Because it's worth doing. Here's a totally new chaperone protein. Let's study how it works. We don't know how it works. Let's characterize it. How big is it? Where are the functional domains? What parts of the protein are important, necessary, or sufficient to help with this folding? How do you study? Is it necessary? This paper was in mice. Is it necessary in humans? Well, it turns out that in 2004, today you would do CRISPR, right? Or you might do hairpins if you were feeling retro. But this was made in an embryonic stem cell knockout mouse model. I asked this person, Bernadette Holdner, for the cells. She refused, saying that she didn't want competitors in her field. And I thought, okay, I'm really stuck, except that was the year siRNA came out. And I had never heard of it. But a friend of mine, whom I was confiding to, told me, why don't you just order an siRNA? They're available at the Whitehead server. They can be designed by computer, ordered, transfected. You can test necessity. It took 2 weeks. That's what I did. Like, I'm not going to walk you through this block, but I'll suffice it to say you can generate an siRNA. I was a fifth-year grad student with no experience in the world of interfering RNAs. I logged into a computer server, ordered an siRNA, ordered a control siRNA against a couple of controls, made a SI-resistant mutant, did this experiment and proved necessity. Because I talked to somebody else about my problem, and I didn't give up on my problem. I had no idea what to do, and then someone reached in and sort of like leveraged me out of the quicksand. So I got this second paper, also in biochemistry, and I went ahead feeling bad, thought, I wasted a lot of time. And I think this paper, it just came out in Science a couple of months ago. It's worth learning about, or worth reading. I think the question is, people are terrified of this word, right? I am. I was, and I'm less so now than I was, and I'm working on being not afraid of it at all. But I am terrified by this notion of failing at things. Instead of, and I have a lesson here where come to a stopping point and you have to redefine the goal to keep going. That's the point, is to keep going. So this is my chairman, Jim Hogle, who is the guy who told me, who is a very famous crystallographer, works on polioviruses, and he's the one who told me, there's more to life than structure. Do the functional characterization. Because, as he told me, look, it's not bad to be ambitious, but you just have to be prepared. And I wasn't prepared. I couldn't have anticipated the absence of a chaperone, but a year in, I should have probably known something was up. And he said, well, you can throw up your hands or you can work the problem. And one of the last things he told me is, don't peak in grad school. The point was to learn how to do this. And the point is to keep asking questions and keep going. That's the point. That's actually not just the point of grad school. To me, that is the point of being in science. And that's the point more largely of being in life at all. There's no thing to get. It's just, can you keep going? Can you keep doing, generating, asking, learning, and becoming something that you're not? And he said also, all of it matters. And it turns out 9 years later, the group that actually solved the structure of this receptor cited my paper and gave me a really nice shout out. Said, thanks to the work of Koduri et al., we understood the requirement of the chaperone. So it mattered that I did the work. And that was really lovely. Yeah. Also, the senior author was my classmate in grad school, which was very humbling. Again. All right, so MD PhDs, finish the PhD, go back to med school. How am I doing on time? I hope I'm not overrunning, but.
Yeah, at least 10 minutes.
Okay. Yeah, we're almost at the end. So I went to med school, then I went to residency. If you're not familiar with it, in the old days, it was a really brutal thing. I did call every 3 days, 4 days if I was lucky, overnight call, 36-hour shifts. We were in the hospital 100 hours a week. And we used every scrap of medical knowledge we had, and we realized, if me at least, I was like, how little we actually know. This was me at the start, looking optimistic, well-rested, and happy. This is me. You can see the creep in my hairline. I've become a little chubbier, but I was the senior chief resident at that point, and my job was to run around the hospital in a suit at night telling people what to do. So, you know, growth, but by the time I reached this point, I had a real sense of modern medicine is amazing and also appalling. Actually, we know almost nothing, and most of what we know is wrong. So it was a real wake-up call. I decided, you can ask me about it if you want, I decided to take a year off residency and the physician scientist track to do something else with my life. And I ended up spending a year teaching medicine in Rwanda, which, as they marked up here, it's about the size of Maryland, about 12 million people. At the time that I went, there were exactly 600 physicians, surgery, medicine, OB, whatever, all told, period, in the country. 600 doctors is not that much, right? So, and this all occurred in the context of the famous 1994 genocide. So rebuilding this country's infrastructure was a mission undertaken by the US government, and they actually recruited young physicians to go over and teach medicine, which is what I did. So I taught medicine in this medical ward, or I gave lectures on a blackboard, and I basically taught all day, 7 a.m. to 7 p.m. For a year. And it was, if I died this moment, I'm a physician scientist. I have committed my life to scientific research. This remains the single unvarnished thing that I am indubitably proud of, and I would take to my grave as a source of pride. Because I think this is the part that gets lost sometimes, science or life or whatever. I don't think we explicitly give credit to this issue. I don't mean in some grand sense like I changed the world for widow remarriage in 1890 in India, or the right to vote for women in this country, it's for suffrage. I mean something as simple as I treated the patient with the right drug. The woman with the arthritis was treated. The person with end-stage breast cancer got morphine. The kids who needed to learn how to do a pulmonary exam did so. Stuff like that. All of it matters. There's no unit for this. And it gave me a sense of meaning that I really wanted to preserve when I left this world and came back to this world. So I came back to Mass General. I became a Hem-Onc fellow, back to the 100 hours a week, now taking care of patients only with cancer, really starting to realize how little we know. And at this time, next generation sequencing had come to the clinic by about this time. So if you came to me with a cancer, you also came with next-gen sequencing of usually like a panel of 300 genes. And I could tell you, Mr. Bob, I hope no one hears me Bob. I'm sorry. But Mr. Bob, okay, fine. Mr. Bob, I'm sorry, you have a mutation in KRAS. Now, I have nothing I can do about that fact. I'm just mentioning it to you as something we know, but it is a useless fact. It's an orphan fact. There is no therapeutic implication for it. But like everything else, right? One step at a time. Knowing was one thing, and especially for transcription factor DNA binding mutations, amplifications, deletions, translocations, loss of function mutation are key to the pathogenesis of this cancer. And those proteins are, as I know because I used to be a structural biologist, they're large. They're mostly unfolded until they hit DNA. They're multi-protein subunit complexes, so a lot of their drug binding pockets are sequestered in interfaces with other proteins. There's nowhere you can put a drug on those proteins, which is why they are deemed classically undruggable. And when you have a mutation in there, up till 2014, we would say, that's too bad. Except that in 2014, this paper came out. And this paper both changed my life, it's given rise to a whole new field, and it's what I do for a living now. And it's what my life's work is. And that is the discovery that this protein, Icaros, it's the master transcriptional regulator of multiple myeloma, the bone cancer. Without this protein, myeloma cells die. It's an absolute requirement. And it turns out that there's this drug called lenalidomide, thalidomide, lenalidomide, other derivatives. You've probably heard of it with some really bad connotations. We can talk about that. It's a great story. But that drug basically tags the protein And the combination of the protein and the small molecule creates a composite, a chimeric recognition surface that can now be recognized by an E3 ligase, an enzyme that will tag it with ubiquitins. And when tagged with ubiquitin, it goes to the proteasome and it's destroyed. And in the absence of this protein, I told you the cell dies. This drug took multiple myeloma from being, if you came to me with myeloma, I would say, you'll live for about 5 years, you'll get an auto stem cell transplant, and then we'll have to see what we do, i.e. palliation. Now we tell people for some subtypes of myeloma with these drugs, something else is going to kill you. Like your heart disease, your drinking, your bad driving, something else. But something else. You have a cancer that maybe isn't curable, but you can turn it into a chronic disease. That's a huge change. And just as we've done for IMiDs with myeloma, the question is, can we do it for anything else? There's a million transcription factors out there that cause cancers. Why don't we apply this technology to that idea? I suspect I'm out of time, so I'm not actually going to tell you what we do. I'll tell you the lab that I went to work for is this guy, William Kalin Jr., who, as you might remember, won the 2019 Nobel Prize. He's the guy over here. He's smiling here. He actually almost never smiles. He's an absolutely terrifying hard ass of a guy, but he smiled for this portrait. And he's famous for working out, along with Ratcliffe and Semenza, worked out the biology of oxygen sensing in eukaryotes. And it turns out that this transcription factor, HIF1 alpha, hypoxia-inducible factor, normally, in a normal cell under normoxia, it's being degraded all the time by the proteasome. And so Bill has some experience with the proteasome. And the idea is that when you're hypoxic, an oxygen sensor tags the HIF1 alpha with a little tag that makes it immune to the proteasome, and now you build up the protein and you turn on your oxygen-dependent program, like more red cells, more blood vessels, or whatever. So this proteasome degradation idea was sort of in Bill's mind anyway, and it's how he sort of got to this field of how did this drug, can we make drugs out of this kind of idea? And so these are the last two slides. I'll say, I won't tell you about the exact science that I did in Bill's lab. Suffice it to say, I took that idea of lenalidomide to Icaros in myeloma, and I've broadened it out to, much like Malcolm would have done, I created a platform and a set of tools, genetic and biochemical, so that for any transcription factor you give me, I can give you a list of drugs and genes that control its abundance. And somewhere in that set, first of all, it's a great research tool, because if you don't know what the protein does, you turn it off and see what happens. But really, if it's a target that really matters to a cancer cell, that's a potential set of therapies. And I do it in leukemia stem cell transplant, because that's where my other life is. These are some famous quotes from Bill that I'll leave you with. He's very famous for saying all of these, and I find them a little shticky, but unfortunately, like all things shtick, there's some truth in it. My favorites are this one, be your own worst critic. And I think you can take that too far, but it is actually important. I like this one, positives beat negatives. You can never do too many experiments. And I love this one. This one is, I think, the one I wish you would take away with. No matter where you're at, what you're doing, the definition of clarity is you have to know what is the question. Because most times I hear talks or I listen to people and they're sort of like getting lost, I always want to say, time out, what's the question? 15 words or less, what's the question? And it helps clarify what you're trying to do. And so I'm going to skip all the sciencey stuff and just say I got to go to Sweden. That's the paper that came out. I got to wear that fancy white tie getup. That's Bill the day he won the prize, just for science nerdism. And now I run a lab. This is me. This is my lab. I run a lab that studies protein degradation. I have a couple of postdocs, a couple of techs. It's a very small lab, but we're in a larger department. We collaborate with everybody we can think of. We go to tea with people in the evenings because we need to learn about what other people are doing. When people ask me to do any talk anywhere, talk to any students, I always say yes because I always learn something. And I hope that's a little bit of a result of both the fortune I had in having the mentors that I did, and I mean that both in terms of Bill was a very difficult person to work for. People left his office crying. One postdoc actually had a heart attack the week before she met with him because the stress was so bad. I have any number of stories about this. But sometimes, the question is, why are we doing this? And anybody watch Ted Lasso? Really, just you and me? You do too. Okay, have you seen that? It's a few episodes. Okay, so this is stolen from that because I also read, for fun, the letters of Van Gogh to his brother, which if you've never read them, it's worth reading. That man struggled in obscurity forever, and he wrote these really heartrending and beautiful letters to his brother about toiling for greatness in the shadows of obscurity. And I love this quote, that you can't expect out of life that you're going to give you things that maybe it can't give you. Maybe the point of living is not the reaping, but the sowing. Maybe the point is to put out things that maybe you'll collect on, but maybe the point is to sow the seed, and that's it. To learn, to grow, to try things, and leave the outcomes and the wins, to leave, that's not what the point is. This is my father's mentor in medical school. His name is Raghuveer Bhagavat. He was a Yale-trained physician who moved back to India at independence. In 1947, he packed up his practice in New Haven and said, I'm going to move to independent India, and I'm going to help teach a new generation of people. And my father was his prize pupil. And he was a what is the question kind of guy. He was a what's the differential? What are the tests? Let's make a plan. Here's our logical answer. And he really inspired, he really believed, he told my father, you have it in you to go to the United States and make a career as an academic physician. And that's me. He did not predict any greatness for me. But he mentored my father in ways that changed my father's life and led him here. I am only here because of that. And I think about that a lot, that I have never had a chance to speak with him. He died, when I was very young. I've never had a chance to sort of say, you sowed the seed in my father, and I am the result. It's kind of an amazing thing. All right, so this is what I learned from Bill at the end. Nothing is ever wasted. There really are no shortcuts. I'm 45 for a reason, and my hair is quite gray. Getting the mentors you need is really hard. And it's not obvious. Bill's a terrible person. You would never want to have a drink with him. It's super anxiety provoking to go into the room. Steve, my PhD mentor, was not the nicest person either. But they also taught you what you needed to know. And there is an aspect of mentorship that you have to be able to say, this is not about me, and I'm going to take myself out of the equation for a second. I want to learn what there is to be learned here, because I care about the larger mission is what I want. And I need to learn whatever there is here, I need it. I need to take that and move on with it. I think about my family a lot. They are, even the ones who have passed away, but the history that you have in you is the momentum that brought you here. And it's yours. It's yours personally. It's just for you. So it's hard not to be afraid, I think, in science and in life. I love this quote too. I think good science, bad science, stupid science, it's all hard. So try to find the big question and really don't be afraid. Get stuck in on it. And doing is always better than thinking. So be an empiricist, do stuff and iterate what you learn, keep moving. And then just for all points, this is my puppy, who is super cute. All right. Anyway, it's not meant to be preachy, it's meant to be, I hope you got something out of it. I've made quite a few mistakes getting here, so I'm hoping that something in there is useful for you. Thank you. Yes, sir.
First off, thank you for your talk. It's great. I'm sending you back to the exam you took in India,
Oh, yeah.
and where you placed, and where you are now.
Right.
And asking, has that shaped how you see potential in others? Right. So you have potential that isn't realized. I'm British, very familiar with that whole system, and where you end up versus where you could have ended up, based on different opportunities.
Oh, yeah.
Has that shaped how you kind of interact with people?
For sure. So most of the postdoctoral opportunities that I get, as a junior person, as a starting person, for example, somebody who has 3 papers in science is not going to apply to my lab for a job. Because if they had any sense, my budget every year, my lab budget per period is about 150 or 200,000 a year. If you have that kind of background, you're an idiot to work for me. you would go to work for my boss, Bill Kalin, because his budget on a Howard Hughes National Academy, yada, yada, is $2 million a year. So I don't see those candidates, right? And I think, again, there are people who go into this job that I'm in right now who say, oh, I can't get any good people. I have to labor, I have to get along with these swabs, But I would say that, I've interviewed now this year probably 10 or 12 people. I've hired 2 people from India. And I feel that what is absolutely clear to me is that within the constraints of the system they're in, like in a country where electrical supply isn't guaranteed, freezers go out, you're like, mix up your stuff in DI water. Oh, guess what? There's no DI water. Do your NMR in D2O. You're talking about make your own D2O. Like, people live and work under experimental constraints that you can't imagine, number one. And number two, therefore, the breadth of the questions they're asking is artificially constrained. And so I read these papers, and in 2 examples that I feel super excited about, they're coming this summer. I am excited both ways. I'm excited for the work, they're gonna work on my projects that I proposed, great. But I'm actually very curious to know, because I firmly believe that moving from that system into our system, I'm not super rich, as labs go, but I have a functioning, well-resourced HMS lab that these trainees will now be able to make use of to take in, and I want them to. I believe that they can take this lab into new directions that I have not anticipated. And I would say that my mentor, Bill, his rule is quite strict. You can only come to the lab if you have a certain quality of paper. He only takes people who went to the Ivies. His technicians have to have gone to the 8 Ivies and BMD, PhD, whatever. He has this world, and it works for him. You can't go wrong in that pool. But I would say it absolutely blinds him to the richness of this issue, right? And the fact that there are brilliant scientists out there that I am so excited to work with. And I think I'm grateful that I spent time in India long enough to understand that. I didn't spend my whole life there. It's different. So I don't pretend to be, like a card carrying. I'm not. But I am someone with some perspective. And it helps. I think it in a good way. Yes, sir?
As a crystallographer, I would love to hear your thoughts about AlphaFold and similar developments.
Mostly I feel irritated. I spent a long time, I remember when I first got access to a robot for seeding, I was so mad. I was like, I spent so much time learning how to be the perfect, how to set a hanging drop tray with exactly 15 microliters of put the wax on and seal the thing. And it was very annoying to find a, I guess I'm a little equivocal on AlphaFold personally. Especially because I work on transcription factors. Like if you put IKZF1, the transcription factor that's the subject of my postdoc in, AlphaFold is very correct at calling, for example, that there are 7 zinc fingers that make up that are in the sequence of the protein. But the first 140 amino acids have never been crystallized. They are intrinsically disordered. AlphaFold doesn't know what to do with that because I don't think people think of it, I think, as like the, DeepMind, or I forget the name of the computer from Hitchhiker's Guide or whatever, like the one that invented the universe as a model for the computer. But it is not that, right? It is using data that exists. And I think it can help you, but it's not magic. I guess that's that's where I'm at. I think I'm actually much more excited about CollabFold. I think where you know the crystal structure of individual partners, and you're asking a energetic question about, are these more likely to interact, or are these more, that kind of a question, I think that's much better, because you're talking about energetic interfaces that you, at least in static form, you understand. Maybe AlphaFold can make guesses about how they might conformationally adjust to one another. So, and then you could port that to, well, if you start adding drugs, so you have a 3-body problem, no one's ever been able to solve those equations in real time, but maybe this is the way. Maybe AlphaFold or CollabFold or something like it can start to show me, when I add lenalidomide to my mix and I'm nucleating this complex, is it just like A plus B plus C like this, or is it that you can start to predict the changes that each molecule made to accommodate the drug to form a new ternary structure, that conformational plasticity. So I'm more excited about that, I guess, than I am about AlphaFold. but it comes up a lot, right? Because fewer structures than we would like.
Any questions? Thank you again very much for taking the time.
Finding that.